In the previous video, we have seen how the Mendelian rules can be applied to the populations, and also how the population can diverge from the Mendelian calculations. What can be the reason that the population does not obey the Mendelian calculations? And now we are going to see how we can do experiments on the population to understand its genetic background. Okay. First of all, there are two terms that you need to know: forward genetics and reverse genetics. As the name implies, the forward genetics was the first to be found, and the reverse genetics came later. Okay. The whole idea about the genetics is to understand the correlation between phenotype and genotype. Because knowing a gene sequence, knowing a DNA piece exists, doesn't mean that it is related to a phenotype. It doesn't make that it makes you smarter, more muscled. You need to understand the variation between individuals in a population and how the DNA molecule directs this difference. So as of now, we know the entire human genome. The Human Genome Project is finished a long time ago, but we are far from understanding the function of each gene or each DNA fragment. So how they work, how they manage to have the phenotype, the trait, we don't know. So to understand that, first people start to get a phenotype, and it's usually a disease, and then they try to find the gene responsible of it. For example, there were many people working with Alzheimer's disease, and they compared the Alzheimer's disease people with the healthy people, and then they tried to understand what is the gene sequence responsible of that. They tried to clone this gene. So back those days, 70s, 90s, cloning such a gene was revolutionizing the studies about a disease, because finally you have the DNA molecule which is which is the director of this process. Okay, people have done forward genetics for a long time. Currently, we are also doing reverse genetics as well. So get a gene and check the phenotype. And how you do that? You first start with a gene which you don't know the function, and then you identify the function. Here is the opposite. First you get a function and you identify the gene responsible of this function. Okay, so we use both forwarded reverse genetics in the field of genetics to make the correlation between genotype and phenotype, which is not an easy job. Okay, many times we do the screens. Genetic screens are highly used. Okay, especially if you read some older articles that is published before 2010 2005, you're going to see a lot of information that is obtained by genetic screens, and currently still we are using the information coming from this data. So how it is done? The idea is this: get an organism, get a model organism, and this model organism should be small enough because you're going to get them in a very high number. So people usually don't do screens in mice because mouse is taking too much place, and these model organisms are very small, usually like C. elegans or flies or yeast or Arabidopsis. And then induce mutations in these organisms, and then check what happens. Now, when you induce mutations, you can do it in several ways. For example, you can use X-ray, or you can use some mutagenic chemicals which harm the DNA and which create mutations. The problem is these assays are so strong, these agents are so strong, majority of the organisms die. I mean, after having that much X-ray, they die, of course, or after having that much DNA breaks, they die. So you get the minority which survives, and these survivors, they usually have some mutations. And these mutations, of course, have some effects. So what do you do? First, when you have a mutation, the first thing is that the mutation is most possibly in the intergenic sequence. Because not only in human, but also in the other organisms, majority of the genome is the intergenic non-functional region, and only the minority of the genome is made of genes. So the mutation you induce is most possibly intergenic region, so it doesn't have an effect, so it doesn't change anything in the organism. That's why it survives. Okay, we don't care about this. Some of them have mutations in the genes, and they have some phenotypes. They can have any kind of phenotype. For example, if you're talking about flies, maybe some doesn't have eyes, or some have no wings, some have wings but cannot fly, some have strange colors, some cannot mate, some are stupid, maybe they forget everything all the time, so they have very low intelligence, they cannot solve problems, something. You can check any kind of phenotypes, and you check these phenotypes, you know the mutations, correlate them. This is what a screen is called. This is how a screen is made. Of course, you have to start with a very large number of organisms, like thousands, 10,000s, hundreds of thousands, to get something meaningful. And it's a very long job. It's really like running a marathon. You have to be very, very patient. It takes like a few years. And at the end, you're going to have any kind of mutations. Some mutations will have substitution, insertion, deletion of, or substitution of multiple nucleotides. Or, by the way, these are called indels, insertion, deletion. Or depurination, deamination, anything can happen. In the next step, you identify the phenotype and you check the mutation, or you find the mutation and you check the phenotype. You can do both of them. In this case, for example, the screen is done on the C. elegans animal, and this C. elegans, normally they feed together because they are social animals, so when they are eating, they eat together. After the screen, they identified a mutant. Of course, these are random mutations because X-ray or the mutagenic chemical randomly damages the DNA. And one of the mutations, they are no social at all. They are not eating together. They don't like each other. So what is the mutation that caused these animals not to stay together, but to stay far away from each other? So go check the mutation, and voila, you're going to find the gene responsible of social feeding. I wonder if it has a counterpart part in humans, and if it works in the same way. Okay, so this is how the screens work. And previously, or currently, we know the function of many genes, and majority of our information, they come from these screens. So from scratch, without even knowing anything about the situation, without having no idea about the genes, they randomly induced these mutations, and they identified the phenotypes, and with this correlation, we obtained so many information. Okay, then there are some newer methods of these screens, which are RNAi screens. RNAi, you all know, is RNA interference, and it's a way to knock down a gene. Okay, it is not knockout, because knockout is something when you remove the gene from the genome, so the gene is not there anymore, you change the DNA, it is called knockout. When you don't change the gene, when the gene is transcribed normally, but it cannot be translated because you are inhibiting the translation by an RNAi, it is called a knockdown. So knockout and knockdown, they usually have the same phenotype because all in all, they don't have the functional protein product. But knockdown is usually very easy. Get this multi-well plate, and in each of them put some C. elegans. The C. elegans are really small, so they're going to fit. They're going to fit in these plates, and on each of them, put some RNAi. And these RNAi are randomly designed. They are going to randomly knock down some genes. So check the phenotype in each of them. By the way, you can do something very nice in C. elegans. Normally, when you're going to use C. elegans on a mammal, you have to change the genome. You have to create some transgenics, or in the flies too, so that the RNAi is going to knock down the target gene. In the C. elegans, you can edit as if you're adding salt. So these added RNAi, they are going to be eaten by the C. elegans, and they are going to be knocked down. They are going to knock down the target mRNA and see what happens. For example, in this case, this is wild type, no knockdown. In this case, you knock down one of the genes, one of the mRNAs, and the organism is sterile. It cannot mate anymore. So voila, you identified a gene which is important for reproduction. Okay, by the way, this is all you can say. You cannot say anything else. To say other things, you have to do other experiments. And another trick that we use in genetics is the conditional mutations. Okay, sometimes, especially when you do knockouts, it is very difficult to keep them alive, because when the organism doesn't have the gene, it easily dies. It's very weak, it is highly affected, the phenotype accumulates. We don't like it. We like that the organism keeps healthy until we induce them. So for example, here you see the heat shock example. These are temperature sensitive mutants. 23 degree is the permissive temperature. So here you don't observe the phenotype, and 36 degree is the restrictive temperature. Only when you heat the yeast to 36 degree, you're going to see the effect of this mutation. Okay, this is called a conditional mutant, and conditional mutants are very useful because they are going to keep the organism alive until you do your experiment. Okay, you can do this temperature sensitive thing in several ways, like using a temperature sensitive promoter or temperature sensitive mRNA, or I don't know, some other binary systems. But all in all, they are very useful. Now, complementation test. So this is a new concept. Please listen it carefully. It is important. Now, in the classical genetics, imagine that you're fishing. Most of the fish you have, they are normal, straight, but some fish you have, they are strange and they are bent. They look absurd. And your neighbor is also going for fishing, and your neighbor also got a fish like this. And as a geneticist, you wonder, okay, I've got this fish, my neighbor got a fish like this, but are they strange because of the same mutations, or are they strange because of different mutations? What you do is that, what you do is that you keep the fish you have got from the sea and the neighbor got, and mate the two. When they have children, if the children look normal, it means that these two fish are complementing each other. It's because the fish you get is strange because it has a mutation in the gene A. And the fish that your neighbor got is strange because it has a mutation in gene B. And these two are different genes. And we know that for the same condition, there can be different genes. I mean, very different mutations can end up in the same phenotypes. Think about dwarfism. If you block the calcium metabolism, it causes dwarfism. If you block the skeleton producing cells from dividing, it causes dwarfism. If there is another problem, it causes dwarfism. There are so many different reasons that people can be dwarf, and each of them originates from a different reason. Okay, here we are talking about two genes, and these two genes can complement each other. The progeny, they are going to be heterozygous for gene A, they are going to be heterozygous for gene B, and they're going to look normal because they're heterozygous for both genes. For each gene, there is a healthy copy, which is going to keep the animal okay. But think that they are the same mutations. Well, here, the alleles are different, A1 and A2, but they can be the same allele too. Mate them, and after you mate them, the progeny still looks very strange. They are still bent, absurd-looking fish. This means that they are the mutants of the same gene. Okay? That means they don't complement each other. If they don't complement each other, it means they are the mutants of the same gene. Okay, previously this was a highly used genetic test, and whenever you check the old data, you're going to see many descriptions that this allele complements this allele doesn't complement this allele. They all come from this kind of experiments. For example, here there is the, I'm not sure which one is female, let's call this male and this female. They are both albino, and when they mate, their children are quite colorful. So I can say that these two mutations are complementing each other because the babies, they have normal pigmentation. So they must be albino for two different reasons. So the mutation they carry must be different from each other. Or you can think about the deaf people. Imagine there is a woman, deaf, and there is a man, deaf, and when they have children, they have several children, and each of them is hearing normally. By the way, this is a commonly seen situation. People meet at the communities of deaf people, and they have normal hearing children. Because you know that hearing is a complicated process. You have to get the sound waves here, and then there are the outer ear, middle ear, inner ear. So it all consists of too many different structures. The sound waves, they should first come here and then pass through these little bones, and then they should travel here. So if you have a mistake here, deaf. If you have a mistake here, deaf, because we know that there's the membrane here. If you have a problem here, deaf. If you have a problem here, deaf. Also, there are some neurons going to the brain. If you have a problem in these neurons, deaf. So maybe their mother is deaf because there is a mutation here in the membrane, and their father is deaf because there is a mutation here affecting the structure. So because the children are heterozygous for both genes, these two alleles are complementing each other, so they don't belong to the same gene. Simple. Yes, new story.